LncPred-IEL, a long non-coding RNA prediction method using iterative ensemble learning. Yanzhen Shu 1, Xiaohan Zhao 1, Shuai Lu 1, Shichao Lu 1, Yanqing Yu 3, Wuhan 1, Leyi Wei 2, 1. College of Informatics, Huazhong Agricultural University, Wuhan 430,070, China 2. School of Software, Shandong University, Jinan 250,100, China 3. School of Mathematics and Statistics, South Central University for Nationalities, Wuhan 430,074, China. Yishu at webmail.herzau.edu.cn, Thou Xiaohan at webmail.herzau.edu.cn, Liushuai_is at 163.com, Skliu at mail.herzau.edu.cn, Yanqing at mail.scuc.edu.cn, Zhangwen at mail.herzau.edu.cn, Wiley at shu.edu.cn. Corresponding author abstract. A large number of transcripts have been generated by the development of high-throughput sequencing technologies. Predicting lncRNA from transcripts is a challenging and important task. In this paper, we propose LncPred-IEL, an iterative ensemble learning long non-coding RNA prediction method. LncPred-IEL not only considers features widely used for the lncRNA prediction, but also take into account sequence-derived features used in the RNA sequence classification, so as to make use of diverse information. LncPred-IEL builds base predictors based on different groups of features, and employs a supervised iterative way to combine base predictors and build ensemble models. Our studies demonstrate that supervised iterative way can learn the representations that help to separate lncRNA and protein coding transcripts, and further improve the performances. Experiments demonstrate that LncPred-IEL outperforms several state-of-the-art methods when evaluated by tenfold cross-validation. The capability of LncPred-IEL for the cross-species prediction is also tested. As complementary to wet experiments, LncPred-IEL is a useful computational tool for lncRNA prediction. Keywords: lncRNA prediction, sequence-derived feature, iterative ensemble learning. Introduction. In recent years, a great number of transcripts has been generated as a result of the development of high-throughput sequencing technologies. 1. Transcripts are DNA translation products, including mRNAs, tRNAs, RNAs and qRNAs. qRNAs are a class of RNAs which are not translated into proteins, and lncRNAs, long non-coding RNAs, are lncRNAs with lengths exceeding 200 nt. Although lncRNAs could not encode proteins, they still play critical roles in various cellular and disease development progress, such as gene express/regulation, 2, gene silencing, 3, RNA modification, 4, and more importantly lncRNAs have been identified to be associated with many diseases, 5. The comprehensive analysis of these transcripts is of great importance but is also of great challenge, and one of the challenges is identifying lncRNA from transcripts, which is critical for downstream biological function analysis. With the accumulation of transcripts, it is very urgent to develop computational methods, especially machine learning methods, to efficiently and effectively identify lncRNAs. Many computational methods have been proposed in recent years for lncRNA prediction. Typically, these methods utilize traditional binary classifiers to build prediction models based on two types of transcripts: long non-coding RNAs and protein coding transcripts, PCTs. These methods make use of different features, such as codon-related features, ORF-related features, GC-related features, coding sequence-related features, transcript-related feature and structure-related features, and formulate lncRNA prediction as a binary classification task of two types of transcripts, coding transcripts and non-coding transcripts, and design classifiers to build prediction models. Support vector machine, SVM, is the most commonly used classifier for lncRNA prediction, which was adopted in CPC 6, CNCI 7, PLEC 8, lncRScan SVM 9, CPC 2, 10, longest 11, and CP-Pred 12. Random forest, RF 13, is also adopted for the lncRNA prediction models, such as CUM 14, and FELNC 15. Logistic regression, LR, 16 is another commonly adopted classifier for lncRNA prediction models, such as Tridigo et al.'s work, 17, and CP8, 18. In order to enhance performances, several ensemble learning-based methods have been developed, such as TLC-LNC-19, Simopoulos et al.'s work, 20, LncPred, 21. Deep learning-based methods utilize deep learning techniques to build binary classification models, such as lncRNA MFDL, 22, lncRNAnet, 23, and LncKDeep, 24. Existing methods make great progress in lncRNA prediction, but there is still room for improvements. Most existing methods pay attention to the transcript-specified features, but some general RNA sequence-derived features are ignored and still deserve consideration. As far as we know, previous literature rarely adopts general sequence features mentioned in Liu's work, 12, which have many successful applications in the classification of biological sequences other than lncRNAs. The ensemble learning techniques have been proved to be useful for lncRNA prediction, but existing ensemble learning-based lncRNA prediction methods usually adopt simple boosting or bagging strategy, 19 to 21, and more flexible and robust authorized licensed use limited to Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explore. Restrictions apply. 556 ensemble learning strategies are still in demand. In this study, we employed an iterative ensemble learning long non-coding RNA prediction method, abbreviated as LncPred-IEL. LncPred-IEL considers many reliable transcript-specified features as well as general sequence-derived features, to make use of diverse information about transcripts. LncPred-IEL builds initial base predictors based on the different groups of features and utilizes a supervised iterative way of combining base predictors to generate new high-level base predictors, and finally build an ensemble model for the lncRNA prediction. Compared with several benchmark methods, LncPred-IEL produces better performances, achieving AUC score of 0.9601 on human dataset and 0.9711 on mouse dataset, when evaluated by tenfold cross-validation. The experiments demonstrate that features learned in a layer-wise iterative way can moderately improve feature representation ability, thus differentiating lncRNAs from coding RNAs more accurately in feature space. In conclusion, LncPred-IEL provides an effective way to alleviate the redundancy of features and enhance the ability of feature representation, and it is promising for the lncRNA prediction. 2. Materials and methods. Datasets. To conduct the experiment, we collect two types of transcripts, lncRNAs and protein coding transcripts, PCTs. GenCode 25 is a comprehensive repository containing gene annotation about human and mouse genome by computational analysis, manual annotation and experimental validation. The annotated lncRNAs and PCTs are downloaded from GenCode as the raw data. To alleviate sequence redundancy, we employ an open-source program CD-HIT 26 to cluster sequences and remove similar sequences with a threshold of 80%. Then, we obtain two types of RNA sequences, lncRNAs and PCTs for mouse, release M21, and human, release 29. We keep all lncRNAs as positive instances, and sample the same number of PCTs as negative instances, and then compile our human dataset and mouse dataset. Notably, the sequence length of two datasets ranges from 9 nt to 91,668 nt. For further evaluation, we adopt CP-PRED datasets 27 about human, Homo sapiens, mouse, Mus musculus, fruit fly, Drosophila melanogaster, and zebrafish, Danio rario, which contain coding RNAs from NCBI Ref-seq 28, release 95, and lncRNAs from Ensemble database, 29, release 90. Table I. The dataset used in this paper: main dataset human 24162, 24162, mouse 27595, 27595 CP-PRED dataset Human 23,384 23,384 Mouse 15,345 15,345 Fruit Fly 2775 17399 Zebrafish 6840 15534 The details about the datasets are shown in Table I. Our datasets are adopted as the main datasets to evaluate the proposed methods and compare different methods. CP-PRED dataset is used for parameter setting and the cross-species lncRNA prediction. The overview of LncPred-IEL. This work presents a long non-coding RNA predictor using the Iterative Ensemble learning, abbreviated as LncPred-IEL. Fig 1 demonstrates the critical components of LncPred-IEL. First, we consider 6 groups of sequence-derived features to encode sequences into feature vectors. Second, we adopt the random forest as the classification engine and build 6 individual RF-based predictors respectively using 6 groups of features. Third, we adopt an iterative feature representation learning framework to generate an ensemble model from 6 base predictors, and then use it as a new base predictor, which is then combined with 6 base predictors to build another ensemble predictor. In this way, the new ensemble predictor is iteratively generated and then added for the next round of ensemble learning until the stop criterion is met, and the final ensemble model is adopted as the lncRNA predictor. When predicting input sequences with the score greater than 0.5 is predicted as lncRNAs, otherwise PCTs. See sequence-derived features in this section. We briefly introduce features for the sequence representation. We consider transcript specified, which have been proved to be useful for lncRNA prediction. Besides, we consider 5 groups of sequence-derived features including spectrum profiles, mismatch profiles, reverse complement MER, pseudo-nucleotide composition and autocross covariance. Fig 1. The flowchart of LncPred-IEL authorized licensed use limited to Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 557 To facilitate the description of features, a given sequence is defined as S equals 1, where is the total length of the sequence and denotes the TH position of the nucleotide in the sequence. Transcript specified features for lncRNA prediction. The features widely used in the related works are considered. The recent studies have demonstrated several features are of the most importance. ORF length, ORF coverage, Fickett score and hexamer score used by CP818. Similarly, ORF integrity, isoelectric point, pi, gravity and instability index are used by CPC6 and CPC230. The composition, transition and distribution, CTD, are used by CP-PRED-12. Therefore, we adopt these transcript-specific features to build our prediction models. The ORF length is the length of the first ORF. The ORF coverage is the ratio of longest ORF length and transcript length, and the ratio of the length of first ORF to the transcript length is also included in candidates. The ORF integrity means whether the longest ORF starts with a start codon and ends with a stop codon. The Fickett score is transformed from the nucleotide position frequencies and base composition of a transcript by a lookup table. Hexamer score is calculated based on in-frame hexamer frequency of coding and non-coding transcripts, and a positive hexamer score indicates a protein coding transcript. Pi, gravity and instability index are structure-related features. Pi means the theoretical isoelectric point of the predicted peptides which is encoded by given PCTs. Gravity and instability index are the grand averages of hydrophobicity and the stability of the predicted peptide. The composition, transition and distribution, CTD, denote the global transcript sequence descriptors. The nucleotide composition denotes the percent composition of each nucleotide along the entire sequence, which is mentioned in Fickett score. The nucleotide transition describes the percentage frequency of conversion of 4 nucleotides between adjacent positions. Finally, 5 relative positions, 0, 25%, 50%, 75% and 100%, for each nucleic residue along the whole sequence is used to denote the nucleotide distribution. Table 2. 6 groups of features: feature dimension parameter, transcript specified, ORF length 1, no parameter, ORF integrity 1, no parameter, ORF coverage 1, no parameter, Fickett score 1, no parameter, hexamer score 1, no parameter, pi 1, no parameter, gravy 1, no parameter, instability 1, no parameter, CTD 30, no parameter, general spectrum profile 1 mer 4, no parameter, 2 mer 16, no parameter, 3 mer 64, no parameter, 4 mer 256, no parameter, 5 mer 1024, no parameter, mismatch profile 3 m, mismatch profile 64 meters, the maximus matches 4 m, mismatch profile 256 meters, the maximus matches 5 m, mismatch profile 1024 meters, the maximus matches, reverse complement K mer profile 1 rifkmer 2, no parameter, 2 rifkmer 10, no parameter, 3 rifkmer 32, no parameter, 4 rifkmer 136, no parameter, 5 rifkmer 528, no parameter, pseudo-nucleotide composition PCs DNC general PCs TNC general SCs DNC General 16 plus 6 SCs TNC General 64 plus 12 ranks DNC Autocross covariance DAC 36 lag distance between residues TAC 4 lag distance between residues spectrum profile. The spectrum profile, also called MER feature, is widely used in bioinformatics. 14 to 16. MER is to count the occurrences of length contiguous subsequences. Given a sequence S, spectrum profile is defined as equals 2, where is the occurrence frequency of corresponding length contiguous subsequences. Reverse complement MER, RIFKMER, profile. This feature takes the reverse complement of an RNA sequence into regard. Given a sequence S, the reverse complement length contiguous subsequences will be cut after generating MER. Then the occurrences of the remaining length subsequences are extracted to constitute a feature vector named RIFK. For example, if equals 2, there are totally 16 basic MERs, AA, AC, AG, AT, CA, CC, CG, CT, GA, GC, GG, GT, TA, TC, TG, TT, but by removing the reverse complementary MERs, there are only 10 unique MERs in the reverse complementary MER feature: AA, AC, AG, AT, CA, CC, CG, GA, GC, TA. Detailed information of this feature can be found in 31, 32. Authorized licensed use limited to Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 558 Mismatch profile. The mismatch profile, 31, is also similar to MER profile but has a parameter less than, for mismatch tolerance in the length contiguous subsequences. For equals 3 and equals 1, the symbol, 3, 1, denotes a 3-length subsequence has a max 1 mismatch. Suppose there is a subsequence ACG satisfy the 3, 1, we need to consider 3 cases, XCG, AXG and ACX. X can be replaced by any nucleic acid residue. Mismatch profile is represented as equals 3, where indicates the occurrence frequency of the TH length contiguous subsequence with mismatches, equals 1, 2, 3, 4, equals 0, 1, 2. Pseudo-nucleotide composition. Pseudo-nucleotide composition considers sequence order information, particularly the long-range or global sequence order effects, via the physico-chemical properties of its constituent nucleotides. 33. In this study, different pseudo-nucleotide compositions are considered, including the basic feature pseudo-dinucleotide composition, SDNC, and 4 variants: parallel correlation pseudo-dinucleotide composition, PCS-DNC, parallel correlation pseudo-trinucleotide composition, PCSINC, series correlation pseudo-dinucleotide composition, SCS-DNC, and series correlation pseudo-trinucleotide composition, SCS-TNC. Autocross covariance, 33, 34, measures the correlation between any two physical chemical properties by transforming the nucleotide sequences with different lengths into fixed-length feature vectors. Autocross covariance has two components, autocovariance, AC, between the same property, and cross-covariance, CC, between two different properties. AC measures the correlation of the same property between two nucleotides separated by a distance of along with the sequences. CC variable measures the correlation of two different properties between two nucleotides, also separated by. In our study, each nucleotide sequence is represented as autocross covariance vector which combines AC and CC. There are several types of autocross covariance feature, such as DAC, dinucleotide autocovariance, DCC, dinucleotide cross-covariance, TAC, trinucleotide autocovariance, and TCC, trinucleotide cross-covariance. Finally, we adopt DAC, the combination of DAC and DCC, and TAC, the combination of TAC and TCC 33, 34, as our autocross covariance feature. The iterative feature ensemble learning method in this paper, a supervised iterative learning strategy is utilized to learn the feature representations from different groups of features to construct the ensemble learning model LncPred-IEL. The construction of LncPred-IEL has three steps: feature optimization, base predictor construction, and generating the new base predictor by ensemble learning. Details are described as follows. First, we have 6 groups of features which contain diverse information about sequences, and we use them to encode the sequences into feature vectors. There are some high-dimensional feature vectors, which makes it necessary to reduce feature redundancy and noise. Here, we adopt a 2-step approach of conducting feature selection for each group to determine discriminative feature subsets. The first step is to sort all features according to their importance assessed by ANOVA, analysis of variance, 35, then the minimal redundancy maximal relevance, MRMR, method, 36, is employed to determine the optimal feature subsets. Therefore, we obtain 6 groups of optimal feature subsets for further analysis. Second, linear prediction is taken as a binary classification problem. Random forest is a machine learning algorithm for classification, regression and other tasks, which has been extensively used in bioinformatics, 37 to 40. We adopt random forest as the classification engine to build predictors because of its high efficiency and high accuracy. The Python package, scikit-learn v.0.20.3, 41, was utilized to implement random forest classifiers. After encoding sequences using one group of optimal feature subset, we can build a prediction model using random forest, and then use it as a base predictor. Since we have 6 groups of optimal feature subsets, we build 6 base predictors. Third, an iterative ensemble learning approach is used to develop the final prediction model, inspired by layer-wise way of learning features in deep neural network, DNN, 42. We combine 6 base predictors to develop an ensemble model. In the stage of prediction, each of base predictors can assign a score or probability for a given sequence, and how to make the final decision is quite important. Ensemble learning is a machine learning paradigm where multiple learners, for example classifiers, features and data, are trained and combined for a task, and it has lots of successful applications in bioinformatics, 43 to 46. There are different ensemble learning strategies for combining outputs from multiple base predictors, such as averaging voting, weighted voting and nonlinear combination. Because of the complicated correlations between different features, we adopt a nonlinear combination approach to integrate outputs from multiple base predictors. For a given sequence, we have 6 scores generated by base predictors, and we combine them to form a 6-dimensional feature vector for the sequence. The 6-dimensional feature vectors are used as input vectors, and the real labels, lncRNA or not, of these sequences are adopted to build a random forest-based ensemble model. Further, the ensemble model is taken as a new base predictor, and added into the set of base predictors. In an iterative way, the ensemble model is generated from base predictors and then used as a new base predictor. The iteration process will continue until achieving max iteration times or observing no performance improvement. 3. Results and discussion. A performance evaluation metrics. The prediction models are evaluated using the 10-fold cross-validation, 10CV. In a fold of 10CV, the dataset is split into 10 equal-sized subsets. One subset is used as the testing set, and 20% of other sequences are kept as the validation set and 80% are used as the training set. The prediction models are trained on the training set and are optimized on the validation set, and then makes predictions for the testing set. The training validation testing procedure is repeated 10 times until each subset has been used for testing. The performances of prediction models authorized licensed use limited to Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 559 are evaluated by several commonly used metrics: sensitivity, SN, specificity, SP, accuracy, ACC, and the AUC score, the area under the ROC curve. These metrics are defined as equals plus equals plus equals plus 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 plus, where TP, true positive, is the number of positive samples predicted to be positive, TN, true negative, is the number of negative samples predicted to be negative, FP, false positive, is the number of negative samples predicted to be positive, FN, false negative, is the number of positive samples predicted to be negative. The ROC curve is plotted by using the false positive rate, 1 SB, against the true positive rate, SN, for different cutoff thresholds. We treat AUC as the primary metric, because it assesses the performance of prediction models regardless of any threshold. B parameters setting for features as summarized in Table 3, several sequence-derived features, such as mismatch profile, SDNC, PCST general, PCSTNC general, SCSDNC general, SCSAINT general, DAC and TAC have parameters, which may influence the performances of prediction models. Therefore, it is critical to determine the parameters for these features. Here, we build the individual groups of RF prediction models on the balanced CP-pred human data, determine optimal features according to the AUC scores of corresponding models. To obtain more information and reduce redundancy, we choose the parameter equals 1, 2, 3, 4 and 5 respectively for the spectrum profile feature, and merge all of them as the spectrum profiles. The same setting is adopted for the reverse complement spectrum features. In the mismatch profile, the parameter is same as MER, while the parameter means the max mismatch tolerance. In this study, we suppose that does not exceed 1/3 of the length of MER. So, we employed 3, 1 mismatch profile, 4, 1 mismatch profile and 5, 1 mismatch profile as the whole mismatch profile subset. The pseudo-nucleotide composition features, SDNC, PCSDNC general, PCSTNC general, SCSTNC general, SCSTNC general have two parameters, where 0.1 0.9 and denotes the highest counted rank of correlation. For SDNC, PCSTNC general and SCSDNC general, 1 2, for the PCSTNC general and SCSTNC general, 1 3, is the shortest length of sequences, and equals 9 in our datasets. We use a grid search strategy to test the combinations of two parameters in the above ranges. According to our results, 5 features, SDNC, PCSTNC general, PCSTNC general, SCSTNC general and SCSTNC general achieve highest AUC scores when using the parameters 7,0.5, 7,0.7, 6, 0.7, 7, 0.1, and 6,0.1, respectively. The autocross covariance features, DAC and TAC have a parameter, which ranges from 1 2, and DAC and TAC produce the best AUC scores when equals 7. Therefore, all parameters are determined for those features, and then sequences are encoded into feature vectors for the following modeling. See performance of LncPred-IEL. In this section, we evaluate the performances of LncPred-IEL on the main datasets using tenfold cross-validation. LncPred-IEL takes an iterative way of constructing ensemble learning models from base predictors and then use them as new base predictors. Here, we analyze the iteration process and discuss how the iteration process can enhance the lncRNA prediction. Analysis of iteration round. As discussed above, the iterative process of LncPred-IEL will continue until meeting the iteration round. To investigate the iteration process, we analyze the tenfold cross-validation performances of LncPred-IEL models in the iteration round from 1 to 50. Results on main human dataset and main mouse dataset are demonstrated in Fig 2. LncPred-IEL produces the best AUC score of 0.9601 on the human dataset in the 18th round of iteration and produces the best AUC score of 0.9711 on the mouse dataset in the 18th round of iteration. In general, the performances of LncPred-IEL will increase as iteration continues, and stay unchanged after reaching a peak. Results demonstrate that the interactive process improves performances in a stable way. LncPred-IEL achieves the AUC scores of 0.9601 and 0.9711, the accuracy of 0.9008 and 0.9134 on main human dataset and main mouse dataset. Fig 2. AUC scores of LncPred-IEL models in the iteration. The x-axis represents iterating times, the y-axis represents the AUC scores of LncPred-IEL. Iterative process enhancing prediction performances. In this study, we adopt 6 groups of sequence-derived features to construct the iterative ensemble learning model LncPred-IEL. Here, we discuss the correlation between 6 groups of features over different species, on main datasets. 6 groups of features lead to 6 base predictors, and their scores for all instances in the tenfold cross-validation are adopted to calculate the Pearson correlation coefficient, which measures the correlation of different groups of features. As shown Fig 3, there are higher correlations between spectrum profiles, reverse complement spectrum profiles and mismatch profiles than that others, because of their same intrinsic characteristics. In general, all groups of features are highly correlated, indicating that the intrinsic correlation between these features may cause redundancy and lead to overfitting of models. Moreover, the main human dataset and main mouse dataset share the same correlation pattern, and this demonstrates that the sequence characteristics between human and mouse are probably conserved. Authorized license to use limited to, Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 560 Fig 3. Heat map of the correlation between the 6 feature types in different species. Although there are high correlations between features, how the iteration process contributes to performance is still unknown and need to be investigated. In the first round of the iterative process, scores from 6 base predictors are used as 6D input vectors to generate an ensemble model and used as a new base predictor. As the iterative process continues, higher dimensional inputs vectors are generated for building the ensemble model. We take the results on main mouse dataset as an example. The final ensemble model LncPred-IEL are constructed on the 24D input vectors generated from base predictors. Large VIS 47 is a visualization tool for large-scale and high-dimensional data. We take the 6D input vectors in the first round of iteration and the 24D input vectors in the last round, and adopt large VIS to show how they classify 2 types of sequences. Fig 4 displays the distribution of positive samples and negative samples of human dataset in 2D feature space. Obviously, 2 classes of samples are not clearly classified by the initial 6D vectors, and positive samples are separated from negative samples after the iteration. The results demonstrate that the features learned in an iterative way can better distinguish positive samples, lncRNAs, from negative samples, coding RNAs, and that is why the iteration ensemble learning in a supervised layer-wise way could enhance the performances. Cross-species prediction. It is challenging and interesting to test the capability of LncPred-IEL for the cross-species prediction. We train LncPred-IEL models on the main datasets of human and mouse, and then make predictions for the two species from CP-Pred datasets, i.e. fruit fly and zebrafish. As shown in Table 3, two pre-trained models perform well on fruit fly and zebrafish benchmark datasets. Specifically, LncPred-IEL trained on main human dataset produces AUC score ranging from 0.8248 to 0.8999 for fruit fly and zebrafish respectively. LncPred-IEL pre-trained on main mouse dataset achieves 0.9395 and 0.9841 of AUC scores for zebrafish and fruit fly, respectively. That is, LncPred-IEL has the ability to distinguish lncRNAs from coding RNAs in a cross-species manner. Fig. 4. Large vis visualization of features vectors, initial feature vectors and feature vectors learned after iteration, for two types of sequences. D. Comparison with other state-of-the-art methods. To demonstrate the advantages of LncPred-IEL, we adopt several state-of-the-art methods for comparison, including CP8, CPC2, longdist, and CP-Pred. Those lncRNA prediction methods report good performances, and are widely used in bioinformatics. All prediction methods are evaluated using 10-fold cross-validation. First, we compare LncPred-IEL with benchmark methods using the main datasets of human and mouse, which include the same number of lncRNA sequences and PCTs. As shown, authorized licensed use limited to Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 561 in Table 4, LncPred-IEL achieves better performance than other methods in terms of all metrics, and significant. Table 3. Performances of LncPred-IEL and the cross-species prediction main human fruit fly, CP-Pred, 0.4288 0.9779 0.9023 zebrafish, CP-Pred, 0.7299 0.9034 0.8506 main mouse fruit fly, CP-Pred, 0.8566 0.9764 0.9598 0.9841 zebrafish Pred 0.7996 0.9522 0.9058 0.9395 Improvements are observed on both main human dataset and main mouse dataset. LncPred-IEL achieved 3% or higher improvement of AUC score on main human dataset, and improves AUC by 2% or higher on main mouse dataset. Notably, LncPred-IEL achieves better SP score than other models on two main datasets, indicating that LncPred-IEL could reduce false positives in the iterative process. To test the robustness of prediction models, we also compile imbalanced datasets of human and mouse, which have twice, 3 times, of the number of negative instances as positive instances. As shown in Table 4, all prediction methods produce better AUC, SP and accuracy on the imbalanced datasets than on balanced datasets, which are caused by the imbalanced data. SN, also known as recall, decrease on the imbalanced datasets, indicating that less real lncRNAs are identified. There are several possible reasons why LncPred-IEL performs better than other models. On the one hand, 6 groups of sequence-derived features bring diverse information. On the other hand, a layer-wise iterative ensemble learning method provides an efficient way to combine informative features. Table 4. Performances of prediction models on balanced and imbalanced dataset human CP 0.927 to 0.859 to 0.7841 0.8297 0.8904 0.9165 0.8784 0.8801 0.8833 0.9399 0.9419 0.93 0.398 CPC 20.9346 0.8513 0.6776 0.8115 0.8789 0.9198 0.8730 0.869 0.8597 0.9342 0.9377 0.9266 CP-PRED 0.8717 0.8761 0.8762 0.7940 0.7925 0.7866 0.8328 0.8203 0.8089 0.8898 0.8905 0.8870 longest 0.7776 0.7926 0.7906 0.7516 0.7466 0.7460 0.7646 0.7618 0.7571 0.8356 0.8413 0.8406 LncPred-IEL 0.9316 0.8702 0.8084 0.8703 0.9114 0.9268 0.9008 0.8978 0.8968 0.9601 0.9590 0.9566 mouse CP8 0.9441 0.8887 0.8399 0.8539 0.9083 0.9253 0.8990 0.9019 0.9040 0.9540 0.9563 0.9557 CPC 2 0.9427 0.8683 0.8223 0.8441 0.8992 0.9288 0.8934 0.8890 0.9022 0.9505 0.9520 0.9544 CP-PRED 0.8773 0.8791 0.3797 0.8035 0.8040 0.6211 0.8405 0.8291 0.65 0.80 0.8876 0.8886 0.5013 longest 0.8842 0.8892 0.8913 0.7907 0.7897 0.7874 0.8374 0.8229 0.8133 0.8047 0.9071 0.9052 LncPred-IEL 0.9345 0.8854 0.83020 0.8927 0.9365 0.9507 0.9134 0.9195 0.9203 0.9711 0.9722 0.96684 Conclusion. lncRNA prediction is a very important topic. This paper presents LncPred-IEL, a long non-coding RNA prediction method, to accurately predict lncRNAs. One of the highlights of LncPred-IEL is that not only does it adopt commonly used transcript-specified features, it also adopts general sequence-derived features, including spectrum profiles, mismatch profiles, reverse complement MER, pseudo-nucleotide composition and autocross covariance. Another highlight of LncPred-IEL is that it utilizes an iterative ensemble learning scheme to combine 6 groups of features. Experiments demonstrate that LncPred-IEL can separate positive instances and negative samples by learning representations in an iterative way, which further improves the performances. Compared with the other 4 state-of-art lncRNA methods, LncPred-IEL produces better results, and its capability in the cross-species is also tested. In conclusion, LncPred-IEL is a useful tool for lncRNA prediction, complementary to experiments and traditional techniques. Acknowledgement: this work is supported by the National Natural Science Foundation of China, 61,772,381, 61,572,368, National Key Research and Development Program, 2018 YFC 04079004, Huazhong Agricultural University Scientific and Technological Self-Innovation Foundation. The funders have no role in study design, data collection, data analysis, data interpretation, or writing of the manuscript. References: 1. J. A. Reuter, D. V. Spacek, and M. P. Snyder, High Throughput Sequencing Technologies, Mole Cell, Volume 58, Number 4, pages 586-97, May 21, 2015, DOI 10.1016/j.molcell.2015.05.004. 2. L. Yang et al., lncRNA-Dependent Mechanisms of Androgen Receptor Regulated Gene Activation Programs, Volume 500, Number 7464, p. 598, 2013. 3. A. T. J. C. O. I. P. B. Wierzbicki, The Role of Long Non-Coding RNA in Transcriptional Gene Silencing, Volume 15, Number 5, pages 517-522, 2012. 4. N. Liu, M. Parisian, Q. Dai, G. Zheng, C. He, and T. J. R. Pan, Probing N6-methyladenosine RNA modification status at single nucleotide resolution in mRNA and long non-coding RNA, Volume 19, Number 12, pages 1848-1856, 2013. 5. S. Chung et al., Association of a Novel Long Non-Coding RNA in 8q24 with Prostate Cancer Susceptibility, Volume 102, Number 1, pages 245-252, 2011. 6. L. Kong et al., CPC, Assess the protein coding potential of transcripts using sequence features and support vector machine, in English, Nucleic Acids Research, Volume 35, No. Supple_2, pp. W345-W349, July 2007, DOI 10.1093/NAR/GKM391. 7. L. Sun et al., Utilizing Sequence Intrinsic Composition to Classify Protein Coding and Long Non-Coding Transcripts, Nucleic Acids Research, Volume 41, Number 17, pp. E166-E166, 2013. 8. A. Li, J. Zhong, and Z. Zhou, Plek, a tool for predicting long non-coding RNAs and messenger RNAs based on an improved K-mer scheme, BMC Bioinformatics, Volume 15, Number 1, p. 311, 2014. 9. L. Sun, H. Liu, L. Zhang, and J. Main, lncRScan SVM, a tool for predicting long non-coding RNAs using support vector machine, PLOS 1, 10.1371/journal.pwn.01396554 Volume 10, Number 10, p. E01396554, 2015. 10. Y. J. Kang et al., CPC2, a fast and accurate coding potential calculator based on sequence intrinsic features, Nucleic Acids Research, 10.1093/NAR/GKX428 Volume 45, No. W1, pp. Authorized Licensed Use Limited to, Lanzhou University. Downloaded on June 1, 2020 at 16 hours and 9 seconds UTC from IEEE Explorer. Restrictions apply. 562 W12 W16, 2017. 11. H. W. Schneider, T. Real, M. M. Brigido, M. Walter, and P. F. Stadler, a support vector machine-based method to distinguish long non-coding RNAs from protein-coding transcripts, BMC Genomics, Volume 18, Number 1, p. 804, October 18, 2017, DOI 10.1186/S128640174178178-4. 12. X. Tong and S. Liu, CP Pred, Coding Potential Prediction Based on the Global Description of RNA Sequence, Nucleic Acids Res, February 11, 2019, DOI 10.1093/NAR/GKZ087. 13. L. Bryman, Random Forest, Machine Learning, Volume 45, Number 1, pages 5 to 32, 2001, DOI 10.1023/8:10109334044324. 14. L. Hu, Z. Shu, B. Hu, and Z.J. Liu, Cum, a robust coding potential calculation tool for lncRNA identification and characterization based on multiple features, Nucleic Acids Res, Volume 45, Number 1, p. E2, Jan 9, 2017, DOI 10.1093/NAR/GKW798. 15. V. Wucher et al., Fionk, a tool for long non-coding RNA annotation and its application to the dog transcriptome, Nucleic Acids Res, Volume 45, Number 8, p. E57, May 5, 2017, DOI 10.1093/NAR/GKW1306. 16. A. Kukiyara, Applied Logistic Regression, Technometrics, Volume 34 3, p. 2, 2012, DOI 10.108000401706.1992.104A5291. 17. F. C. P. V. M. P. G. Tradigo, on the identification of long non-coding RNAs from RNA-seq, presented at the IEEE International Conference on Bioinformatics and Biomedicine, BIBM, 2016. 18. L. Wang, H. J. Park, S. Dasari, S. Wang, J. P. Coker, and W. Leyi, CP8, Coding Potential Assessment Tool using an alignment-free logistic regression model, Nucleic Acids Res, Volume 41, Number 6, p. E74, April 1, 2013, DOI 10.1093/NAR/GKT006. 19. J. Hu and B. Andrews, Distinguishing Long Non-Coding RNAs from mRNAs Using a Two-Layer Structured Classifier, presented at the 2017 IEEE 7th International Conference on Computational Advances in Bio and in Medical Sciences, ICOBS, 2017. 20. C. M. A. Simopoulos, E. A. Wertelnike, and G. B. Golding, Prediction of Plant lncRNA by Ensemble Machine Learning Classifiers, BMC Genomics, Volume 19, Number 1, p. 316, May 2, 2018, DOI 10.1186/S1286401846665-2. 21. CPN et al., LncPred, Classification of Long Non-Coding RNAs and Protein Coding Transcripts by the Ensemble Algorithm with a New Hybrid Feature, PLOS 1, Volume 11, Number 5, p. E0154567, 2016. 22. X. N. Fan and S. W. Zhang, lncRNA MFDL, Identification of Human Long Non-Coding RNAs by Fusing Multiple Features and Using Deep Learning, Molecular Biosystems, Volume 11, Number 3, pages 892 to 897, 2015. 23. J. Beck, B. Li, S. Quan, and S. Yoon, lncRNAnet, Long Non-Coding RNA Identification Using Deep Learning, Bioinformatics, 2018. 24. C. Yang et al., LncADeep, an ab initio lncRNA Identification and Functional Annotation Tool based on Deep Learning, Bioinformatics, 2018. 25. J. Harrow et al., GenCode, the reference human genome annotation for the ENCODE project, Genome Res, 10.1101/gr.135350.111 Volume 22, Number 9, pages 1760-74, 2012. 26. L. Fu, B. New, Z. Ju, S. Wu, and W. Leyi, C. D. Hit, Accelerated for Clustering the Next Generation Sequencing Data, Bioinformatics, Volume 28, Number 23, pages 3150-3152, 2012, DOI, 10.1093/bioinformatics/BTS 565% J Bioinformatics. 27. Xton and S. Liu, CT Pred, Coding Potential Prediction Based on the Global Description of RNA Sequence, Nucleic Acids Research, 10.1093/nar/GKZ087 Volume 47, Number 8, pp. E43 2019. 28. N. A. O'Leary et al., Reference Sequence, RefSeq, Database at NCBI, Current Status, Taxonomic Expansion, and Functional Annotation, Nucleic Acids Res, 10.1093/nar/GKV1189 Volume 44, No. D1, pp. D73345, 2016. 29. D. Arzerbino et al., Ensemble 2018, Nucleic Acids Res, 10.1093/nar/GKX1098 Volume 46, No. D1, pp. D754 D761, 2018. 30. Y. J. Kang et al., CPC2, a fast and accurate coding potential calculator based on sequence intrinsic features, Nucleic Acids Res, Volume 45, No. W1, pp. W12 W16, July 3, 2017, DOI, 10.1093/nar/GKX428. 31. S. Gupta, J. Dennis, R. E. Thurman, R. Kingston, J. A. Stamatoyanopoulos, and W. S. Noble, Predicting Human Nucleosome Occupancy from Primary Sequence, PLOS Compute Bio, 10.1371/journal.pcb.100013 Volume 4, Number 8, p. E10000134, 2008. 32. W. S. Noble, S. Kuhn, R. Thurman, M. U. and J. Stamatoyanopoulos, Predicting the In Vivo Signature of Human Gene Regulatory Sequences, Bioinformatics, 10.1093/bioinformatics/P1047 Volume 21 Supple 1, pp. I33843, 2005. 33. W. Chen, T. Y. Lei, D. C. Jin, H. Lin, and K. C. Cho, SKNC, a flexible web server for generating pseudo-tuple nucleotide composition, Anal Biochem, 10.1016/j.ab.2014.04.001 Volume 456, pages 53-60, 2014. 34. Q. Dong, S. Zhou, and J. Guan, a new taxonomy-based protein fold recognition approach based on autocross-covariance transformation, Bioinformatics, 10.1093/bioinformatics/BTP 500 Volume 25, Number 20, pages 2655-62, 2009. 35. J. R. G. Miller, Beyond ANOVA, 1997. 36. H. Peng, F. Long, and C. Ding, Feature Selection Based on Mutual Information, Criteria of Max Dependency, Max Relevance, and Min Redundancy, IEEE Trans-Pattern ANALMOC Intel, Volume 27, Number 8, pages 1226-38, August 2005, DOI, 10.1109/POMI.2005.159. 37. L. Luo, D. Li, W. Zhang, S. 2, X. Ju, and Ji Qian. Accurate prediction of transposon-derived RNAs by integrating various sequential and physicochemical features. PLOS 1, Volume 11, Number 4, p. E0153268, 2016. 38, W. Zhang, J. Shi, G. Tang, W. Wu, X. Yue, and D. Li, Predicting Small RNAs in Bacteria via Sequence Learning Ensemble Method, 2017, IEEE, pages 643 to 647. 39, S. Liu et al., LncGF Stack, a global sequence feature based on a stacked ensemble learning method for predicting likeness from transcripts, genes, Volume 10, Number 9, p. 672, 2019. 40, W. Zhang, X. Yue, G. Tang, W. Wu, F. Huang, and X. Zhang, SPL LPI, Sequence-Based Feature Projection Ensemble Learning for Predicting lncRNA Protein Interactions, PLOS Computational Biology, Volume 14, Number 12, p. E1006616, 2018. 41, F. Pedregosa et al., Scikit-Learn, Machine Learning in Python, Journal of Machine Learning Research, Volume 12, No. October, pages 2825 to 2830, 2011. 42, Z. H. Zhou and J. Feng, Deep Forest, R14 preprint R14:1702.08835, 2017. 43, L. Wei, S. Wan, J. Guo, and K. Wang, a novel hierarchical selective ensemble classifier with bioinformatics application, ArtIntelMed, Volume 83, pages 82 to 90, November 2017, DOI 10.1016/j.artmed.2017.02.005. 44, L. Wei, P. Singh, J. Zeng, J. Chen, R. Su, and F. Guo, Improved Prediction of Protein-Protein Interactions Using Novel Negative Samples, Features, and an Ensemble Classifier, ArtIntelMed, Volume 83, pages 67 to 74, November 2017, DOI 10.1016/j.artmed.2017.03.001. 45, L. Wei et al., Iterative Feature Representations Improve N4-methylcytosine Site Prediction, Bioinformatics, 10.1093/bioinformatics/BTZ408 2019. 46, W. Zhang et al., SFLLN, a sparse feature learning ensemble method with linear neighborhood regularization for predicting drug-drug interactions, Information Sciences, Volume 497, pages 189 to 201, 2019. 47, J. Tang, J. Liu, M. Zhang, and Q. Mei, Visualizing Large-Scale and High-Dimensional Data, presented at the proceedings of the 25th International Conference on World Wide Web, WWW 16, 2016.